This talk is now going to be actually specifically about a package, namely MDAnalysis, and we are very happy to have one of the maintainers of the package here, Irfan Alibay. Thanks for making it to SciPy and telling us all that is new about MDAnalysis.
Okay. Well, thank you so much for inviting for giving us the opportunity to present our work on MDAnalysis. Just, I guess, a little housekeeping. I will be putting these slides up on Zenodo later. And also, we're a very sort of cat-obsessed project. So if you see any images of cats, I just want to sort of highlight that these were all made by Fiona. And, you know, it's always great to have a little bit of art in your talk. And there we go. So I'm going to start by doing the acknowledgements. This is a package that has been around for 17 years. I've not been there for most of the time, and honestly, the work really is down to all our contributors. We've had 206 contributors as I've last checked, and many community members that have helped along the way through issues or discussions. And really, none of this would be here without them. So. This is all to say that I'm sharing some work and I'll be highlighting some contributors, but you know, all of this is only possible because of everyone's contributions to the work. So hopefully to many of you in this room, this might not need saying, but I'll go through sort of a little bit of the problem statements to why we have MDAnalysis and you know, going a little bit on what the type of simulations we look at. So I think the main statement to make here is that molecular structure is key, or understanding molecular structure is key to understanding function, particularly when it comes to atomistic resolutions. So here we have an example of a transporter. This is a sodium glucose transporter, also known as SGLT, which allows the movement of glucose across a cell membrane. And the main take-home message here is that when this transporter allows for glucose to cross the membrane, it undergoes some kind of sort of reorganization of its protein. So it goes from sort of an inward-facing to an outward-facing state. And the message I'm trying to convey here is that by understanding how the protein reorganizes itself as it transports a sugar, we're able to understand the function and sort of the underlying biology of how sugars are being transported across cell membranes. And this is quite important because then it allows us to then understand how we can either hinder or help that process along, and then maybe develop new therapeutic solutions. And so just understanding how structure changes at atomistic resolution is sort of the key driver here, and it is something that isn't just limited to biomolecular systems. It's really all types of systems. We saw it in the previous two talks. Understanding, for example, how materials rearrange themselves help us then eventually build new and better materials. And so the question is, how do we achieve this? So experimentally, we can get good information about atomistic details of molecular systems, particularly biomolecular systems. We've had lots and lots of great work on this. You know, there are various methods like X-ray crystallography, NMR, cryo, and so on that can get really good, accurate models of biomolecular systems. And you can see the PDB is a good example of this. The PDB is essentially, you know, having I guess maybe an exponential explosion in new entries every year, and to the point where we are now able to train these really cool machine learning models, such as AlphaFold, where we can then predict protein structures in ways that, you know, for systems that we can't even look at right now, and that's amazing. However, one of the issues we have with experimental data is that it is in some ways limited. Yes, with something like NMR, you can get some kind of time-based resolution, but it's usually, you know, the distance between your frames is usually quite high, and it doesn't really give you the full picture. And so being able to understand time-dependent rearrangements, like the ones that we saw in the SGLT case, is still important to do, and we need to be able to somehow see that. We also can't really easily see environmental interactions. You can sometimes resolve some lipids or some waters in an X-ray crystal, but you really can't really understand how, for example, solvent moves around a protein. And finally, and probably more important, in my opinion, is you can't really quantify thermodynamic properties that easily. Yes, you can do other experiments that will help, for example, you can do some sort of affinity measure if you're trying to understand how a ligand binds to a protein. But that's not always accessible, and it's sometimes quite expensive. And so we want to be able to have a means by which we can get this type of information. So how do we overcome this? As a computational chemist, I tend to turn to simulation to be able to enhance experimental data and effectively eventually derive the properties that I mentioned in the previous slide. And here I am mostly going to concentrate on the idea of molecular dynamics simulations and particularly apply to standard molecular mechanics force fields. So these are, the type of system, very sort of crude and maybe not exceedingly well detailed ways in representing a system. It's not QM level, but it is still very useful for the type of work we do when we look at very large systems. So here's a kind of a rough overview of how this works. So on the far right, we have effectively a series of potentials that define our system. So the way we do this is we define a system as a series of spheres, which are our atoms, that are connected together by bonds that essentially act as springs. And then we fit some types of harmonic potentials to the way our bonds stretch, the way in which they rotate. We then fit some basic non-bonded terms for how they interact with each other in the longer ranges. Then once we have some sort of an description of our system, which is usually fitted to some higher level of theory, so maybe quantum mechanics data, then we can, using the coordinates and some sort of thermodynamic description of our system, so some temperature and so on, we can then start integrating our forces using sort of classical mechanics, so your standard F equals ma. Based on that, we can then start deriving how our coordinates change over time. So we can create a trajectory of coordinates, which is essentially what we have here at the bottom. And through that, we can see how our systems evolve over time. So here on the left, we have a case of this, where I ran a molecular dynamic simulation of an ADK protein, and the ADK protein, effectively, you can see it sort of open up as you run the simulation. So how does this work in practice? So in practice, if I want to get from simulation to result, I will take in a set of coordinates and some sort of description of the energies of my system. I will find, for example, an NMD engine that I want to run my simulation in. And there are many of these. It could be OpenMM, GROMACS, AMBER, and NAMD, and so on. And it really depends on what I want to do. And then from that, I will run a molecular dynamics simulation, and I will output my trajectory out as some file format. Now, the file format depends on the MD engine I chose, and there is no consistency in most cases, so it will change. And so we have kind of like this mixed bag of different engine and trajectory formats, and then at some point I want to be able to read from these trajectories, get some sort of features, and potentially write a paper about what I found out. And this is where MDAnalysis comes in. MDAnalysis essentially tries to cover the question mark. How do I get from a series of trajectories that I've produced to some kind of set of features that I want to inspect my system, that I want to describe my system? And so MDAnalysis has been around for a while, about 17 years old. I think 2006 when we started. And it's an open source Python library for handling and extracting features from atomistic molecular simulations. I am mostly concentrating on molecular dynamics because that's kind of the mainstay set of simulations that come through our library. But really, we can handle pretty much any atomistic molecular simulation that has a constant number of atoms. So if your number of atom changes, we can't do that right now. It is completely community-led development, and it's engine agnostic. So we can read file formats for most MD engines, and we can represent it internally as a sort of a consistent internal representation of microsystem. So you could read in from GROMACs and LAMPs and be able to do the same analysis and get the same types of results. And the way it works is kind of shown at the bottom. We read in data from a simulation trajectory. We expose it as a sort of we expose the data from the trajectory as a set of NumPy arrays that you can inspect. From that, we then offer a variety of tooling to be able to analyze and process that data, and then hopefully that would then lead you to get some insights and maybe a publication. So in terms of key features, the library, as per its name, has analysis methods. And so we have a variety of built-in analyses. These are your standard distance-based calculations, RMSD, RMSF, the SSB, and so on. We also have a low-level distance library that can help you then build out new analyses if you need to do anything that's custom. We have different ways in which you can sub-select your system, so you can either index your atoms in the way you would a NumPy array, or maybe you want to do some sort of keyword-based representation, so you can, for example, use our select atom methods and for example, say backbone and name CA, and then if you have a protein, it will select the backbone atoms and specifically sub-select your alpha carbons. We have various ways in which to do trajectory manipulation, and particularly on-the-fly transformations. So if you're loading in coordinates and you want to, you have PBC effects you want to deal with, you can, for example, load in coordinates and automatically post-process them to deal with alignment or handle PBC wrapping and so on. And we're extensible. And this is the main point of the library, is that we provide tools for you to do your own analyses. So if you want to create custom readers because you have a new file trajectory format that you want to handle, or new analyses methods, we provide tooling to enable that. So I'll dive a little bit into the organizational library and how it would look like. So the main object model for MDAnalysis is the universe. A universe is essentially a description of your molecular system, offering all the positions and all the chemical information. So in order to create a universe, you would need two things. You would need to know what are your particles. That's offered through some kind of topology file. So we have an I/O interface for reading in topology files, and where your particles are. And then that's through a coordinate file, and we have an interface for that. And then on top of that, then we have this transformations layer, which would then, as you read in from coordinate file, then apply a series of transformations to your coordinates so that they can be handled or be represented in a way that you want them to be. So maybe you want to fix PBC effects as you load in your coordinates. There is a third I/O layer, which is converters. So should you want to read in directly from another library, there is a means by which you can do this. So right now it's limited to RDKit, OpenMM, ChemFiles, and PubMed, I believe. But I'll mention this later, but we're planning to extend this. And the idea is that you could directly read in a molecular object from another libraries and just read that in directly into a universe. So once you create a universe, you would then have your entire system. And so you might want to look at a certain portion of your system. So you would do a selection. And from a selection, you would get a set of atoms that you want to handle or manipulate in some way. And this is what we call an atom group. So an atom group has a series of positions that are loaded in from the coordinate reader every time step of trajectory. And then you can start doing sort of weird and wonderful things with it. So the first thing you could do is write out another file. So in the simplest case, MDAnalysis could be used to transfer from one file format to another. So the more useful side of things, or in some ways the main aim of the library, is then told us to do some type of analysis. So we offer some math utilities and various built-in analyses in order for you to post-process a set of atoms or a selection of atoms in your system and get some kind of results. So you might, for example, look at all your oxygens and your hydrogens and try to find out what parts of your system is hydrogen bonding, and use that for further analyses. So how does this look in practice? So I'll start with a really simple use case, which is RMSF. This is root mean square fluctuation. So the idea is that we have a protein that we've done a simulation of. We want to see how it fluctuates within the length of the trajectory. So very simply, the idea is that you would load in or at least in Python-wise, you would import in all the necessary things. You would import MDAnalysis, you would import your transformation module, and your RMS module from your analysis, the analysis library of MDAnalysis. You would load in your topology trajectory as a universe, so this U equals MD universe, and you would then do maybe some kind of subselection of your system and create these so-called quote-unquote atom groups. So here we can, for example, select out the protein, what's not the protein, and then the alpha carbons, because actually when we do an RMSF, we might want to look at our alpha carbons because the way in which they fluctuate is a good representation of, you know, the backbone or each residue in your protein. And then in this particular case, we've loaded in we have a trajectory that has periodic boundary conditions, and our coordinates have crossed the period boundary. So what we want to do is then apply a set of transformations to fix period boundary condition effects, and also apply some kind of rotational and translational fit, because we're doing a root mean square fluctuation calculation, and what we want to do is only capture movements that are not related to rotational or translational changes. So we apply our transformation, as is shown in the second box, to fix the PBC effects. And then finally, we use our RMSF module, the RMSF class from our RMS module, to calculate an RMSF on our C alpha positions. And it's a single line, and you would get the data necessary to generate this here on the left. So we have a matplotlib generated plot of the RMSF of each alpha carbon along each residue. And then what I've done is I've put that data onto the leftmost representation of the molecule, of the protein, and just to show that, for example, areas that are blue don't move a lot, areas that are red move a lot. And so it's as simple as that. Now you can do far, far more complicated things. So here's an example here of some work that had been done on classifying open and closed transitions of MERDI. And I'm not going to go into the detail, but you know, it is possible then once you've able to extract these features to use, for example, a random forest classifier to be able to understand or cluster out parts of your system. And some of the really cool things that you can do is go beyond just analysis. So this is work done by Brady Johnson, who's been interacting with the MDAnalysis team for the last year or so. And he's done some really cool work with this thing called molecular nodes, where we can then now directly interact with Blender and interface molecular simulations into Blender and create these really amazing movies. And so I would really urge you to go have a look at molecular nodes, because I find this like amazing. So that's a very brief tour of the MDAnalysis library. The library isn't just the whole of MDAnalysis. We have a wider organization. As part of the organization, we have in the core, we have the library and our user guide. We then also have standalone packages. So for example, we have Dystopia that I'll talk about to do fast distance calculations, ways in which to handle grid data. We also have tools, so we have reusable GitHub actions, our Sphinx team, and so on. We have various MDAnalysis toolkits I'll cover, which are downstream packages of the core library. And then we have non-code things such as outreach. And this is really important because that's how we survive. So we do a lot of mentorship for Google Summer Code and other projects. And then also we do workshops. So that's the MDAnalysis organizational library in a 18-minute tour. So the rest of the talk, I'm going to talk about what we've been doing and sort of in the last couple of years and as we move towards a 3.0 release. So as I mentioned earlier, we started in 2006. We had a 1.0 release in 2020, 2.0 in 2021, and we're moving towards a 3.0 in 2025. And the main thing here, and I'll point you to this QR code to look at our roadmap, but the main things that we're really looking at are the bullet points that I'm showing here. We really want to make this library usable for modern and future simulations. So we need to make it a lot faster. We also need to increase the range of applicability. And for us, we also need to make sure that it's maintainable. So we're working towards a slimmer and more maintainable library. We also want to increase our community growth. So that's a last thing I'll touch on. So in terms of performance, the main thing to take home here is simulations are becoming really large. They're much larger than, when I was doing my PhD. We now have GPU-enabled everything, and we're dealing with multi-terabyte data sets for millions of atoms. And we need to do, complex on-the-fly featureization quickly. And right now, what we're happening is we're moving from what should have taken seconds through to minutes and even hours of analysis. And so we need to fix that in some way. So one way we're doing this is by overhauling our data structures. So we're moving through towards some siphonization, and we're doing a lot of work, and particularly Hugo and Richard doing a lot of work in reorganizing data structures so that they're all kind of at the sea level, and there's a lower memory overhead to transferring data to and from sort of faster C API readers. We also are doing work towards improving our distance calculations. So we do have a C library for distance calculations, which are BBC aware, but it's getting old. So Hugo and Richard have worked on this dystopia library, which significantly speeds up distance calculations by leveraging vector instructions. So we can get up to about 10x speed up in certain cases. We also do per frame parallelism. So thankfully, a lot of our analyses are embarrassingly parallel. It's, if you're doing hydrogen bond analysis, it's each frame is independent. So you can do them separately. So work that started with Yushuan in his Google Summer of Code 2-3 years ago. He's now a core developer for us. And Igor last year has been working on how we can sort of hide and bake in multiprocessing and DAS to be able to do parallelism directly. And so they've been able to show that in some cases you can get about 5-8x speed up all the way to the I/O limit. And we're hopefully delivering this at the next release. Then we're also looking to extend our domain of applicability. So this is a variety of things. We want to increase our interoperability for our converters. So Cedric several years back did an RDK converter, and Luna's working on OpenBabel one. And then hopefully we'll have ones for ASC and OpenFF interchange soon. We want to increase our guessers so that we have better sort of chemical information that we can derive from our sort of input files, which is something that I has been working on. And we're also working on new readers. So as you saw before, we're I/O limited now if we really leverage parallelism. And so what that Lawson's doing now this year with Google Summer Code is to leverage ZAR in order to do some really wonderful parallel streaming and remote data access for large data sets. We're also looking to slim the library. So one of the issues that we found is that our contribution model was people will submit pull requests to MDAnalysis, and we would accept the codes and maintain it for them. But it turns out that that's what we found out over the years. That's not a really maintainable approach to things. Having a centralized solution where everyone contributes their code to us really hinders progress. The problem is that we're a few maintainers and we're not experts in everything. And there are lots of people who contribute things. So there becomes this increasingly large barrier to maintaining things. And we, for example, can't really add in new dependencies because then our code becomes bloated. So it becomes really hard to accept new code. So what we're doing actually is, as part of an EOSS4 initiative, we're working on this MDAKits idea. It's based on the similar idea to SciKits. We're asking people to develop their own codes that use MDAnalysis and sit downstream of us. And we're providing them tooling to help them create their own packages. And we're even creating a registry to test it regularly and make sure that it's well integrated, and so that our user base knows where to look for these packages too. And the way it looks is that we have a registry called the MDAKits registry that every week we test all the kits that have been submitted and make sure that they work and tell our users, hey, these work, these do what they should be doing, please use them. And what we've also been able to do is start removing things from our library. So for example, whole 2 MDAKit is being moved out of a library, and that lessens the burden in maintaining the core library, which makes releases a lot easier. And here's an example of various MDAKits that have been developed, so mainly through Google Summer Codes. These are internal, but there's also a bunch of external ones that have been submitted over the last 2 years. And the final thing that I'm going to touch on is non-code stuff. And so Work that we're doing for an EOSS 5 grant is try to help grow our community. And so Jenna, who's our community and project manager, has been doing a lot of work on this and creating community events and doing a lot of outreach through, for example, Outreachy and Station One and Google Summer Code. And the main thing I'm going to highlight here is that we have a UGM in London in August. And if anyone's in the area, please go attend that. And I'll finish by doing another set of acknowledgements to all 206 contributors. And please get involved. Have a look at our codes, have fun with it.
Thank you, Irfan. It's time for questions. Yes?
This might be a bit of a question because I remember I was software engineering and kind of touch on work balance, but as far as I know, like kind of just like Google Maps and some of them have their own capabilities to do RMS, RMSF, and stuff like that. Like, if I remember correctly, you can probably plug it into GPUs and things. So you just give an example. But I guess the question is, like, let's say you're just using Google Maps or something like that. Would it just be better to just stay with it unless you use MDAnalysis?
I first need to repeat the question, so it's, or maybe you want to repeat the question and then you can
please,
answer.
please.
Okay. Yeah. So the question is roughly, some molecular dynamics packages already bring analysis features, and if I stick with one package, am I better to use that, or does MDAnalysis bring something on top?
Yeah, the answer is yeah. Look, when I started, I used Amber and I used the Amber tools, and they worked well. And if all you're doing are basic analyses or the analyses that are available within the package you're using, that's great. The main issue comes when you're trying to do something that's not available, right? In GrowMax, the GrowMax tools are all C++ based. Extending them is not easy. They're all command line tools. And so if you want to do anything additional, if you want to have some sort of dashboard of your results, you need to go somewhere at the Python layer. But yes, I generally like the diversity of tools, so I'm not going to say don't use GrowMax tools. Please do. But if you want to do complex analyses, then you really have to go towards MDAnalysis or something like that.
I'll just go ahead and ask the question. So you showed as an example proteins and the analysis on proteins. But so since this is the chemistry and materials session, I was wondering, are people using MDAnalysis for analyzing materials?
As far as I'm aware, yes. We have several folks that are showing up every so often asking a lot of questions about lamps. We do have lamp support. So I'm guessing the answer is yes. I'm like a pharmacist and biochemist by training, so I don't have any research on it, but the answer would be yes.
Yeah, go ahead.
You mentioned the choice to do SciPy to speed up a lot of the work which you're done with NumPy. I guess, could you tell me a little bit about the design decision to use SciPy as opposed to maybe writing more C++ or maybe using Rust or something like that, numbers around it? Several years ago, I tried to do SciPy. I found it a little bit difficult to find documentation for it.
So the question is, you're using Python for some of your speed improvements. Why use Python and not go lower level and write the C++ or Rust directly?
Okay, I will caveat all this by saying that I am not the one doing the Python code and therefore can't really speak to the entire design decisions. So I mentioned Pythonization, and that's true. We do have some Python for certain things. We do also have some C++ directly for some other things. So it's a kind of a mixed bag of the two. Rust, I think we're kind of staying away from at the moment, mainly because it's not the expertise of anyone on our team at the moment, and also we've not really found a reason Rust over C++ yet. But yeah, at the moment, when I mean Python, I mean a mixture of C++ and Python. It is, particularly when we're looking at these data structures that were in Python already, in some ways it makes sense to just use Python because we can keep it in sort of the structure it used to be at before. I don't know if that answers your question. Okay, awesome.
All right, I think that that means the half hour is up. So let's thank our speaker again.